The Large Hadron Collider, or LHC, is the biggest scientific experiment in the world. An underground ring of high-tech equipment, 27 kilometers long. Within this tube, protons are fired towards each other at near the speed of light. The impact of their collision creates millions of new particles, a sort of miniature Big Bang, and it's the products of these collisions that the scientists are studying. The experiments have already confirmed the existence of a new particle, thought to be the elusive Higgs boson, the key to understanding the formation of mass. And they're set to throw further light on the existence of dark energy, dark matter, and even extra dimensions. The physicists' work is driven by their search for universal, predictive laws. But they're not the only ones who have been searching for fundamental answers. The biologists have also been on a quest for nothing less than the key to life itself. The journey began in the 19th century. Biologists were beginning to ask how the huge variation between Earth's species came about. We'd been breeding specialized animals and plants for thousands of years, well aware that characteristics pass from one generation to another. But no one had connected this with variations in nature, something then assumed to be God-given. When, in 1831, naturalist Charles Darwin set out on a voyage on board the Beagle, he little suspected that his life's work would call such dearly held assumptions into question. But his destination, the Galapagos Archipelago, a chain of volcanic islands cut off from mainland contacts for thousands of years, was to provide him with a perfect example of species variation, ready-made examples crying out for study. The islands had very different ecosystems, and on each one, the animals, though similar, had very different characteristics. After studying the animals of the various islands, he concluded that living things are descended from common ancestors, but not in straight lines. Instead, branches develop over time, each characterized by features most favorable for survival in the environments they inhabit. Darwin knew that when animals and plants reproduce sexually, there can be unexpected variation in the offspring. He reasoned that some naturally occurring variations, for example in the shape of beak among birds, might confer an advantage, and that advantage would result in better survival and reproductive chances. The beneficial variation would thus appear in the next generation, with the process repeating itself, eventually resulting in a new species. When his book On the Origin of Species was finally published in 1859, it presented a powerful case. The idea that humans were linked to other primates in an evolutionary chain initially provoked either revulsion or derision, but Darwin's work could not be seriously refuted. Evolution was a breakthrough in thinking and became the foundation for the most exciting work in biological science. What no one, including Darwin, then knew was how the characteristics of individuals were passed from parent to offspring. Nor could anyone explain how characteristics possessed by neither parent could arise in their young. No one knew what the hereditary mechanism was, where it lay, or how it operated. The unraveling of that mystery was due in part to one of the earliest aids to science, the microscope. As microscopes improved, biologists saw, for the first time, dividing cells. As they observed the staggering complexity of a developing embryo, they realized that they were looking at the fundamental mechanism of new life. They could see that as each cell divided, so did its nucleus. And as the nucleus divided, its contents split evenly in half. It was clear that the nucleus was the critical element, and that it held some sort of blueprint containing instructions for the new cells. Further research revealed that the blueprint was composed of deoxyribonucleic acid, or DNA. Now we had the key to cell replication, but its workings remained a frustrating puzzle until, in the mid-20th century, two British teams made the first all-important breakthrough. Francis Crick and Jim Watson used data researched by Rosalind Franklin to prove that the DNA molecule was a sort of corkscrew shape. It was, in fact, a double helix of two identical strands of atoms. As the cell nucleus split, the molecule's two strands peeled apart, dividing the genetic blueprint exactly. Subsequent research has established that the genetic material is composed of thousands of genes, chemical messengers that define the organism's cells, dictating their shape, development, and function. The groundbreaking Human Genome Project was set up to locate and identify every one of our genes. Initially, the task was performed painstakingly by hand, an almost impossible task. But new computers rapidly took over to complete the process. Now we can pinpoint specific areas of our genetic makeup and even manipulate them if we so wish. Once we know which genes are responsible for what, we can screen for inherited diseases, identify risk areas, and even create therapies for existing gene disorders. It means we can also change the genetic structures of embryos, opening the door to genetic engineering, such as the cloning of Dolly the sheep. We've delved deeply into the nature of matter and probed the secrets of life. Inevitably, they've come together in a new field, nanotechnology. We see the possibility of producing custom-designed biological machines so small that they're on the scale of living cells. They could be DNA or proteins created to perform specific tasks. Fast-multiplying bacteria could convert toxic waste or create unlimited quantities of substances such as oil or drugs. There's already progress. In the US, scientists are using nanotechnology to replicate the process by which plants photosynthesize.
We can grab the machine, the protein, inside the plant called photosystem 1, which is responsible for generating this energy for the plant, and hijack its function, essentially, put it on a substrate of our own choosing, and use the energy to create solar electrical power and to run light bulbs or any kind of machinery that you want.
The resulting substance can be produced in the form of a paint that can be used to coat buildings.
We're domesticating protein molecules, protein living machines, essentially, to do things that are interesting to us. And there's a huge menagerie of such proteins in the natural world, from plants and animals and all sorts of other things, such as bacteria, that can make pretty much anything you want.
The area where nanotechnology may bring the most extraordinary results is medicine. In Montreal, scientists are using modified bacteria to propel miniaturized robots that can move through the human body guided by computer. Once they reach their destination, they deliver drugs to treat just their very specific target. Already, researchers have successfully guided a microchip in living tissue. Now they're working on reducing its size so that it can travel within even the smallest blood vessels. We're pushing the boundaries in all these fields of science, and the implications going forward are profound. Decisions taken now will have major repercussions in the future.
We're beginning to be able to take control of the workings of our minds, our memories, our metabolisms, our personalities, and our kids. We have the means through genetics, robotics, information, and nanotechnology to control matter and energy and life itself at the most basic levels. We've never seen anything like this before, and this is raising profound questions about what it means to be human and what our lives and our futures and our children will be like.
We've learned more about science in the last 50 years than in the whole of human history, with the rate of technical advance now doubling every decade. Progress itself is unstoppable. It is now the job of society to decide which directions to allow our curiosity to take us, and which of the semi-open doors we will push through into the future.